We have progressed from mastering how to repair genetic mistakes to writing our own code. The prospect of us creating new forms of life is now upon us. We've come to realize that DNA is just like software. Again, this has staggering implications. It seems most probable that God designed life, but not humanity. Hi folks, welcome to the 38th episode of the Quintology Files. Today, we shall address something rather fundamental: life. In the previous episode, we discussed the Garden of Eden, traditionally linked to the creation of life. We used to credit God with the creation of life in the myriad forms that we see around us. But as we learned how life on this planet has evolved over the past 600 million years or so, our views have changed. In the same way that it seems unlikely that God created, say, carbon dioxide or hydrochloric acid, the physical evidence does indicate that the miraculous design which enables protons and electrons, the raw material of atoms, which under ubiquitous physical laws applying across our universe, may combine to create a few hundred elements, and these elements further combine to create an almost unlimited range of chemical compounds. This was designed by an intelligence far beyond our understanding. Similarly, it seems we've now realized that there is no basis to claim God created each and every life form. But what if God created DNA in a way that it could adapt to our planetary environment? And to take that one step further, did God design the raw materials of atoms to potentially combine in ways that could evolve into amino acids which might connect up in strings, which could grow into operating systems for self-replicating organisms? What we've come to learn about DNA is truly staggering, with huge implications for how and where life started and for our origins. Today, we shall explore how life works and why it seems most probable that God designed life, but not humanity. This will shock many people, but it is similar to our discussion in an early episode that yes, God designed the complex ways that atoms behave in our universe, but it is equally clear that God did not create the Earth. Our planet was simply an inevitable product of the machinery that God put into motion. Nor is Earth likely to be anything like unique. It is probable that there are uncounted millions of Earth-like planets. Nor is Earth particularly stable or long-lasting. Many people do believe God made Earth for humans to live. Hominoid life on Earth has only been possible for a small fraction of Earth's 4.5 billion year existence. Less than 1/10 of 1% of its present age. And us, Homo sapiens, have only been here for 2 or 3% of that 1/10 of 1% of Earth's existence. And we know Earth could become uninhabitable again very suddenly from a large meteor impact or from eruptions from the gigantic magma chamber swelling yet again beneath Yellowstone National Park. Biology is a way of structuring matter and controlling flows of energy at a molecular scale by slotting each atom into a specified place. Until Charles Darwin developed the theory of evolution in 1875, we had generally assumed that the vast array of life forms on our planet had been individually created by God. Evolution explains how life forms adapt to changing environments. Those with the attributes most suited to particular climatic conditions and food resources will flourish, whilst those less well suited will struggle, summed up as a survival of the fittest. Archaeological and genetic evidence indicates humans first begun to cross-pollinate wild cereals and pulses around 9000 BC, and first begun selective breeding to domesticate goats and sheep around 8000 BC. Chinese voyages of discovery in the 1420s spread chickens around the world, whilst, starting a century later, the Columbian exchange begun large-scale mixing together of the fauna and flora of Eurasia and the Americas. In the past few decades, we have discovered truly staggering knowledge about how life works, how it may be modified, and we've even created its constituent materials from scratch. DNA, deoxyribonucleic acid, is located in the cell nucleus of every cell of our bodies and in every other life form on Earth. DNA is ubiquitous on our planet, providing the genetic blueprint for all organisms, all animals, all plants, fungi, insects, bacteria, and virus. True, some viruses use a simpler form, RNA, but to reproduce, they have to occupy a host cell with DNA in order to replicate.
You say all forms of life on Earth are built using DNA?
Yes, and the implications of that fact are staggering. So, of course, we'll explore it. Moreover, we've come to realize that DNA is just like software. Again, this has staggering implications. As most people are aware, DNA comprises a twisted double helix of information containing about 3 billion base pairs, storing code comprising 4 chemicals: adenine, guanine, cytosine, and thymine, known as A, G, C, and T. Each base pair is linked to a sugar molecule and a phosphate molecule, with the base pair forming a step and the sugar and phosphate forming something like the sides of a ladder. The sequence of these steps, nucleotides, forms the information to build and maintain the specific life form. Each cell in a human being contains an exact copy of its DNA. DNA operates a host of mechanisms using a range of tailored enzymes to check for uniformity and repair any transcription errors when copying DNA during cell division. Differences in DNA between different humans are much less than 1%. Studies have found amazing degrees of similarity between humans and other species. Chimpanzees are closest, sharing 96% of DNA in common with humans. The Abyssinian domestic cat shares 90%. Mice share 85%. Cows share 80%. Zebrafish 70%. Chickens and fruit flies share 60%. And even a banana tree, without any of what we term vital organs, shares about 55% DNA with humans. The shared genetic material found in the zebrafish is so great that 84% of all human genetic diseases can be studied in zebrafish.
How did we come to understand this?
75 years ago, back in the 1950s, we discovered that genes, the instruction sets used to build molecules of proteins, were written on long strands of DNA, resembling stock prices on old-fashioned ticker tape. We learned how each gene specialized in using a combination of amino acids in a specified sequence to make a protein, which was then used by DNA to make the specified parts of a specific organ in a life form. By the 1970s, we started to transfer traits from organisms in which they had evolved to organisms we wanted to exhibit such traits, simply by cutting and pasting sections of the ticker tape. Thus was born the biotechnology industry. In the 1980s, we designed biotechnology machines that could synthesize DNA letter by letter, which were soon used to create wholly new gene sequences not found in nature. Building cells which would work in completely new ways. Effectively, we're reprogramming cells. Soon after 2000, we realized that biology, like computing, is based on digital code, and that cells could be engineered in the same way electrical circuits and software codes are. An amazing advance flowed from the discovery in 2007 of a cellular defense enzyme named CRISPR-Cas9, which cuts up and destroys any foreign DNA that invades a cell. CRISPR stands for clusters of regularly interspaced short palindromic repeats, just in case you were going to ask the question. The Cas9 protein is an enzyme which acts as a pair of scissors, cutting up the attacking DNA and absorbing the alien spacers to serve as a memory bank to identify and fight off similar attacks in the future. It's amazing. Genes encode a series of messages and instructions within their DNA sequences. Gene editing involves changing those sequences, thereby changing the message. This can be done by inserting a cut or break in the DNA, tricking a cell's natural DNA repair mechanisms into introducing the changes wanted. By 2012, CRISPR-Cas9 had been transformed into a simple, programmable gene editing tool.
Has this been used to treat medical conditions?
Yes, procedures were developed which offered the potential to correct genetic defects, such as cystic fibrosis and cataracts. In March 2020, it was reported that CRISPR-Cas9 had been used inside a person's body for the first time to operate directly on DNA to treat a disease. The case involved a patient with an inherited form of blindness, and it was undertaken at the KCI Institute in Oregon, US. The delicate procedure successfully injected through a tube the thickness of a human hair CRISPR-Cas9 to make cuts each side of the mutation and insert correct genetic code. The repaired cells restored the patient's eyesight. Amazing. Gene editing has been deployed to re-engineer seeds to improve crop yields, develop drought tolerance, and enhanced nutritional properties. Genetic modification has been introduced to a population with the intention of altering future generations. For example, introducing sterility amongst female mosquitoes to suppress malaria. These skills have also been used to develop synthetic biology. Biology may be described as the manufacture of proteins which then carry out all the basic functions of life: breathing, digesting food, etc. Each protein is made from a specified selection from 20 amino acids strung together in a chain. The shapes that these chains fold into fulfill different functions as both complex and unpredictable. But we've learned that sequence of the amino acids determines the shape, and the shape determines the function. This is absolutely key and represents an enormous breakthrough for humanity. The gene of a given protein is simply a section of its DNA, a sequence of the 64 three-letter combinations that can be made up from the 4 bases we referred to earlier, simply as A, C, T, and G. These 64 combinations are called codons, and each has a specific amino acid meaning. Each gene is like an algorithm, a set of sequential rules to make a protein. The act of reading the gene, codon by codon, is the act of creating the protein, amino acid by amino acid. We've identified the software code by which cells turn genes on and off. Only when a gene is on, or expressed, will a cell make the protein described by that gene's ticker tape sequence. When it's turned off or repressed, the protein's production stops. Because proteins are the molecules that carry out almost all the tasks that go on within a cell, which genes are expressed and when is fundamental to how cells work. Indeed, almost every form of cancer arises when the ONC gene fails to stop expression of a gene. When genes execute algorithms this way, biology looks like computer science. But biology does not deal with information the way that humans do. In human programs, the logic and the machinery that acts on it are kept separate. Computer users can change a program independently of the hardware that it runs on. But life does not make any such distinction. Biological processing is simply molecules interacting, an extraordinary symphony that can turn a fertilized egg into an entire human. Biological information and operation are fully integrated. Life runs not on software and hardware, but in allware. So the sequence in which amino acids are made determines the shape of a protein that will be produced, and the shapes produced determine the function of what is created. It has been estimated that nature has developed around 5 million proteins, but we only understand the function of a tiny minority. Beyond the 5 million proteins estimated to exist in nature, we have technology that now enables new proteins to be designed at will. Most proteins contain 66 links with 20 different amino acid possibilities for each of the links in the chain. That's roughly the same as the number of subatomic particles in the visible universe. Staggering. So, humankind has discovered how life works. Life is a complex chemical process by which self-replicating organisms evolve and adapt to their surroundings. Is it conceivable that a supreme designer imagined how proteins and electrons could combine to create elements which may then form special compounds which in specified conditions would link up into amino acids? Scientists have managed to recreate these specified conditions in the lab and have seen amino acids form spontaneously. Conditions on early Earth would have been more favorable. Our moon originally orbited much closer, generating much greater tides and an accelerated change from high tide to low tide as it orbited Earth much faster. Weather would have been much more extreme than today, with much more frequent lightning. The spark of energy to fuse compounds swilling around in warm rock pools along the seashore. That was nature's lab. However, it seems it took almost 3 billion years for the first multi-cell organism to emerge on planet Earth. In the next episode, we shall explore how humanity is moving from understanding what might be the way God created life to mastering the technique ourselves. We have progressed from mastering how to repair genetic mistakes to writing our own code. The prospect of us creating new forms of life is now upon us. Thank you very much. I know you can't wait to see the next episode. Please press like and your questions, please. Thank you.